Hello everyone. Hope you all are doing good. My name is BINA TIWARY. I'm working at AcadPrime as a science teacher. Today I'm here by a topic DNA, right? That is a genetic material which we are also calling as an inheritance material. The word itself is indicating that inheritance, that means that we are getting it from our ancestor, that is passing on from one generation to another generation, right? So this genetic material, that is DNA, it is passing on from the grandparent to the parent to the ourselves, and we ourselves will pass on to our progenies. So this is how this genetic material is passed on from one generation to another generation. Now like the full form of this DNA is deoxyribonucleic acid. It is a long polymer of a small single unit nucleotide. These nucleotides are arranged themselves in a multiple time to form a long chain of this DNA molecule. Here you can see the two strands are running parallel to each other. These two strands are running in an anti-parallel direction, that is 5 prime to 3 prime, and other one way from 3 prime to 5 prime. So that is why we are calling it as an anti-parallel direction. And as you can see here, like few, just like a when we are having a ladder, so in the ladder few strands, few in between the joining strands are there. So here also some of the hydrogen bonds are there which are joining these nitrogen bonds. So first of all, let's do a study about the nucleotide. What is that nucleotide is having? So basically this nucleotide is having a sugar molecule and a phosphate molecule, and here a nitrogen base pair, right? These nitrogen base pairs are of a two type, purine and pyrimidine. So purine is having two base pairs, adenine and guanine. This purine will be double helical structure, and here not helical but double ring structure, double pyrrole structure. But pyrimidine is single pyrrole structure, which is thymine and cytosine, right? There are some basic rules are there. Like it's not like any nitrogen base pair can join with anyone. No, it's not like that. Here, adenine will always join with the thymine, and guanine will always join with the cytosine. They will join by making a hydrogen bond. As you can see, adenine and thymine, they are joined together by the two hydrogen bond, and guanine and cytosine are joined together by three hydrogen bond, right? And there will be a very strong affinity for them. Why? Because they are complementary to each other. Like just like a magnet is having south pole and a north pole. They will get attracted toward each other very easily. Just like that, they are having a very strong affinity for them. So these two strands will come together very easily. They will form a hydrogen bond. And this hydrogen bond gives a stability to these DNA strand, because this DNA strand is a very long helical structure, right? So this has to be have some stability. That stability is given by this hydrogen bond. Now this DNA molecule, or better to say like this DNA polymer, they are present in a long strand, right? But they will join together on a octamer. That octamer is histone protein. H1, H2A, H2B, H3, H4. These are the some of the histone proteins are there. DNA, which is acidic in nature, this acidic DNA molecule will make a twist and turn over. Twist and turn, that means they will coil themselves on this nucleosomes. So these nucleosomes are making an octamer structure like this, on which the DNA molecule will make a coiled structure. So what happened? Like this string will be compact in a small space. As you can see, so many nucleosomes are there. These nucleosomes will join together, and then they will give a this tetrad structure, which is known as a chromatid. Each part is known as a chromatid. And when this is during the cell division, before the cell division, there will be a small condensed structure, which we can call them as a chromosome. And when the cell is in the resting stage, so there will be like a thin strand, and this strand is known as a chromatin, right? So this chromatin will have this the nucleotides, and this nucleotides are giving rise to a gene. So all the species like dog, whale, hen, human, all are having different number of a chromosome. Human is having 46 chromosomes are there, right? So now think to yourself, like why always cow is giving birth to cow, rabbit is giving birth to rabbit, we human is giving birth to human. Why it is not like a human is giving birth to a whale? Why it is not happening? Because of this genetic material. The arrangement of this nitrogen base pairs has in such a way that they will make a living organism in the shape what they want. Like there will be a specific arrangement, and this specific arrangement is responsible for the our gender as well as like what we are going to be. So if we will talk in terms of a human being, so like around our 99% of our base pairs are, like as you can see in this slide here, they are saying that the 99% of the base pairs are similar in all the human being, right? In here 99% are same in all people, and in a human around 3 billions of a base pairs are there. So that means this purine and pyrimidine, they like adenine and guanine and thymine and cytosine, they are making a certain type of a arrangement. The 3 billion base pairs are arranged in such a way like all the base pairs are same, but they are their arrangement is different. Like maybe one will have the sequence like this, another person is having sequence like this, right? So these are the two different base pair sequence, and believe me, like these three segment will make a codon, and this codon will responsible for the one of the amino acid that we are having. So this amino acid will again join together to give a protein, right? So whole structure is the will change if one of the base pair will change, so the codons will change, then the amino acid will change, then of course the protein is going to change. So whatever we were thinking, it will all on a different thing, right? So that is the reason like all the base pairs are same to all the living organism, but still all living organism are having different characteristic. Like maybe me and my siblings all are having same base pairs, but some of the characteristics are same. That means some of the genes are passed on from our parent to us in the same manner, but some are having little bit twists and turns. That means a segment of a chromatid, like during the cell division, what happened, like a segment of a chromatid, like suppose this is the chromatid, right? So this when it is passing by to the offspring, so what happened, this will get cut off and this will join as an extra part over here. Now whatever the genetic makeover this P1 is having, P2 will have certain different genetic makeover. So that is the reason like we siblings are having little bit different characteristic, but most of the characteristics are same. Now coming to the another question, like the another question, like we are having some genetic disorders like the thalassemia, hemophilia, sickle cell anemia, and baldness. So why only few people are suffering from these genetic disease? Why not all the human races are suffering from same type of a genetic disease? Because of this change in a base pairs. This change in a base pairs can be done due to the chemical reagents or due to ultraviolet radiation, and that process is called as a mutation, right? So this is a rare phenomena. That is why only few percentage of a human races are suffering from genetic disorder, and that genetic disorder will be passed on from one generation to another generation. And like it's a very good thing, like most of the genetic disorder are reflecting only when they are recessive. So that is very rare chance that both the chromosomes will be having the same type of a recessive characteristic. So we are not having very often that kind of a diseases. So whatever I was trying to say you about the DNA structure, so definitely it's not an assumption it is not that we have done. Some scientists are there which are responsible for the discovery of a DNA. So first lady, she was a lady. She did experiment using using X-rays, and she has given the basic structure of a DNA. After that, some other scientists like Watson and Crick and M. Wilkin, they jointly use the data that is given by the Rosalind Franklin, and then they have given a structure of a DNA. Of course, still now we are following the same trend. They have given a structure of a bDNA. That bDNA is having right-handed, and that is having in between the twist and turn, they are having 10 base pairs. So as you can see here, the base pairs are having some minor and some major grooves are there, and in between this minor and major grooves some base pairs are there, right? This is called as a twist, and in this twist 10 base pairs are there. As you can see, adenine is always joining with the thymine and guanine is joining with the cytosine. Now here in the 10 base pairs are there in a one twist, that is bDNA, and depending upon the base pairs that we are considering for our gene, like as you can see over here, some part of this DNA strands which are running in an anti-parallel direction to each other and are having twists and turns, they are considered to be a gene, and these genes are responsible for the characteristic that we are reflecting. So here we can say this gene is responsible for the characteristic that we are reflecting, and these genes are nothing just a base pairs arrangement. So it is believed that a single chromosome can have around 20 to 25 thousands of a gene. Now you may ask, like, we are not having that many characters, so what these genes are doing? So actually some of the genes are like maybe two or three genes are responsible for one characteristic. Like a simple example I will give you, the blood group, that AB and O. So this blood group is not governed by only one gene. 3-4 genes are responsible for giving rise this characteristic. Same thing, like we are having skin tone color, a variation of a skin tone color is there. That is called as a discontinuous variation. Only not gene, one gene is not responsible for it. So many genes are responsible for it. At the same time, what happened? Like one gene is participating in governing the 3-4 characteristic. So like so many fundas are there that we will cover in our next topic. So what the basic thing I wanted to make you understand, I hope it is clear. So see you in the next video. Have a nice time. Bye.